This video is about anti-cancer drugs that act by binding to DNA. We'll do lots more about transition metal complexes towards the end of year 13. But one drug is Cisplatin. This is a complex of platinum. The complex is formed when a group or ligand with a lone pair forms a coordinate bond to a central metal atom or ion. In this case we have two ammonia molecules and two chloride ions that donate a lone pair of electrons in a coordinate bond to form the complex. This complex is neutral electrically because the two positive charges on the platinum ion are cancelled by the negative charge on two chloride ions. It has a square planar shape, so the two nitrogens, the platinum and the two chloride ions are all in the same plane, flat on the page as I've shown it. We say the coordination number is 4. That means that there are 4 coordinate bonds. Transition metal complexes display isomerism, and this isomer is a cis isomer because the two chloride ions are on the same side of the complex and the chloride platinum chloride bond angle is 90 degrees. The alternative would be the trans isomer, in which the two chlorides would be at 180 degrees to one another. One of the causes of cancer is the uncontrolled division of cells in the body, and this leads to tumour growth. Cisplatin binds to DNA, it stops the DNA being replicated and transcribes, and it stops the cells reproducing and leads to cell death. Because the cisplatin is neutral electrically, it can pass through the nonpolar cell membrane. Inside the cell, the cisplatin is hydrolyzed. That's breaking a bond using water, and the platinum ion chloride ion bond is broken and one of the chlorides is replaced by a water. You can see that this reaction is an equilibrium. In order to keep the equilibrium shifted to the left hand side, the cisplatin is injected into the patient in a saline solution. That's a solution of sodium chloride. You have a high concentration of chloride ions, it shifts the equilibrium to the left. Inside the cell, the chloride ion concentration is much lower, so the equilibrium shifts to the right and the cisplatin is hydrolyzed. Once it becomes positively charged, it's trapped within the cell and can't leave through the cell membrane. Cisplatin works by binding to a guanine residue. On the diagram I've highlighted some of the atoms in the guanine. The three in purple are the atoms or groups involved in hydrogen bonding in the base pair to the cytosine in the opposite strand of the DNA. The nitrogen that I've highlighted in turquoise is the one that the cisplatin binds to. Having bound to one guanine in a single strand of DNA, the cisplatin then reacts with a second guanine in the same strand. You can see in the new complex that the second chloride ion has been replaced by a guanine. So the platinum complex is now cross-linking two guanines in the same strand of the DNA. This cross-linking causes the DNA double helix to kink. Normally the helix is straight, it has a straight axis right up the middle of the DNA. Now the double helix is kinked, and this stops the enzymes that replicate and transcribe the DNA from working, and that's what leads to cell death. It's really important to realise that the cross-link is between two guanines in the same strand of the DNA. It's an intra-strand cross-link rather than inter-strand cross-link. Cancer drugs are very toxic. The idea in cancer therapy is to kill the tumour, but unfortunately cancer drugs also affect normal cells. The cancer drug has most effect on cells that are dividing very quickly, and so the types of cells that are affected are hair cells. That's why people who are having cancer therapy often lose their hair, and also the red and white blood cells, and that's why people having chemotherapy are often anemic and needs treatment to help support their blood. The damage to white blood cells causes immune suppression, which makes chemotherapy patients susceptible to infections. When companies are developing a new drug, they have to prove that the drug is safe and more effective than available treatments. And it also needs to be shown to be cost effective. The organisation that regulates drugs is called the Medicines and Healthcare Products Regulatory Agency, and NICE controls access to the drugs under the NHS, whether the NHS will pay for a new drug. People having chemotherapy, the doctors will discuss the potential benefits and adverse effects of drugs with patients. We try to minimise the effects of chemotherapy by using the lowest possible dose that works. Cisplatin is quite an old drug. Newer cancer drugs are being developed which target tumours more specifically. So instead of affecting all the cells in the body, they just affect the tumour. Lots of innovative strategies have been used to do this. Quite often drugs have been linked to antibodies that just bind onto the surface of the tumour, or you can use lasers to activate the drug. So there's lots of different ways in which pharmaceutical companies have developed more specific drugs that have fewer adverse effects.